cases we have to choose a more strict than a more relaxed penalty gap? So this is a very good question. So this is a good question because it is hard to have an answer, but I have an idea and I have some sort of experience on where to select more strict penalty gap. So we should know that there is no sort of sequence-based optimization for the penalty gap. So for the best practices, alignment tools come with some sort of a default value for gap penalty, but it is sort of optional to change it. And how do you understand whether you want to change it or not? So if you sort of increase the penalty score for gaps, it means that you don't really tolerize, sort of, you are not tolerant to any gaps. If you decrease the gap penalty score, then it means that you are more tolerant to the mismatches, right? So it is some sort of a balance here. So it depends on your interpretation and your protein family or DNA family of interest. And if you just look at the alignment and there is a biological reason for you to have less or more gaps, then you can play with it. You can increase or decrease the gap penalty. However, there is, as far as I know, there is no tool changing the gap penalty for you. So it's a custom or manual process that you have to apply. But what cases you choose? The cases where the gaps are more than they are supposed to be, then you can change it. Or when in the cases when you think that there should be gap rather than mismatches, then you sort of decrease the penalty score for the gaps. I hope it answers Manos's question. All right, let's move on. Okay, so as I said, so far we just said that, okay, +1 for match, -4 for mismatch. However, we can be sort of better than that. We can have better substitution scores. So I'll give you one example. For protein alignments, we have amino acids, right? Some amino acids are more similar to each other than the rest. So for example, aspartic acid and glutamic acid, they look very similar, except for one sort of carbon group. They are almost identical, right? So these three groups are also very similar. So, and in real life, we know that these two amino acids can replace each other often. If this is the case, so okay, if glutamic acid replaces itself, it is the best score. However, D is not a bad option as well. So that's why its score should be high also, maybe not as high as DE, but DE substitution should also have higher score. Because in nature, if you look at nature and protein sequences of organisms, you will see that they replace each other often. So that's why there are some sort of popular substitution matrices offered by first Dayhoff, which is the, I would say, pioneer of bioinformatics, and BLOSSOM matrices also offered, which are used in BLAST algorithm. So, and BLOSSOM is very popular and it is still used. So this is how it looks like. So if glycine and glycine matches with each other, its score is 6. And for example, proline is 7, it's higher. So one thing that is sort of interesting is tryptophan, which is 11. So when tryptophan matches to itself, its score is the highest. So do you have any idea why this would be? So for example, glycine, alanine is 4, tryptophan is 11. Why would that be? Do you know? Or does any of you have any idea? You can write to chat or you can speak up if you like. Okay, I will come to that. So if you also look at some of the values, they are negative. So what does it mean? This phenylalanine, whatever it matches with, it will have sort of unfavored substitution, except for neutral ones, which is leucine and isoleucine, which is L and I here, right? So this is a symmetric sort of matrix here we are talking about. So let's come to that tryptophan issue. So it is 11. So the reason why this is 11 is tryptophan is a rare amino acid. It is the rarest. So that's why when you find tryptophan and it matches itself, then it is most likely, that it's a true match. So therefore, it is 11. On the other hand, alanine is a frequent amino acid. So that's why its value is lower than tryptophan. Okay. So now we have our blossom matrix. So what do we do with that? And okay, before what we do with that, I would like to show you, at least mention about what sort of scoring matrices we can use. As we said, identity matrix is used. So it means that if there's a match 1, there is no match, it is 0. So it's simple as that. However, it's not that useful. Mutation data matrix. So Blossom and PAM, how do we define or how do we derive them? Or how people derive these matrices is the question. Is it just basically just looking at the chemical structure and the physical chemical properties of amino acids? No, it is not actually. So if you look at here, these are physical properties of amino acids. You can also derive from this matrix, or these properties you can derive a matrix. However, this is not used. And genetic code, so amino acids are coded based on the similarities in the sort of codons. You can also derive a matrix from that information. However, it is also not used. What is used is mutation data matrix. So as you can infer from the name, the source of these matrices are not physical chemical properties. They are from mutations. They are real observations. So as we said, we have homologous sequences, right? And we have our genomes available. So what we do is we just look at the variety of species. We look at the substitutability of amino acids across the sort of tree of life, I would say. Then from this mutation data, you can infer which amino acids are more likely to substitute each other. And this is where we have PAM and Blossom matrices derived from. So finally, I have to talk about heuristic methods, including BLAST. So we have to have sort of heuristic methods because of the complexity. It's a large scale to cover everything. So that's why, so instead of Needleman-Wunsch or Smith-Waterman algorithm for local alignment, BLAST algorithm is some sort of different. And what it does is it breaks sequences into words. Okay, and after that, it just matches for the perfect match. It just finds the perfect matches. Then it scans the database for that. And after that, it tries to extend that match. Okay, so it is sort of fast, and also it is sensitive, but it's not as we would like it to be. Sometimes it cannot find the removed homologies, and we may have a chance to talk about it too. So BLAST, I'm sure you are all familiar with, but have you ever thought about its algorithm? So first the step is to mask the low complexity regions. Why? So low complexity regions, from the name you can also infer the complexity, the variety of amino acids for some regions are low. So these regions are called low complexity regions. Why they are sort of pain is because you don't really know which lysine, which is K, is aligned with which one, right? And it messes things up. So you don't want them, and they are not informative at all. So what we do is we remove the low complexity regions from the analysis. Then we can add them later on. After you align, you can add them, whatever. So it's not a big deal. But these are, first, they don't give any information. And second, they can sort of mislead you. So that's why there's a mask for the low complexity regions. And these regions can also be dispensable. For example, these chicken, zebrafish, they don't have these low complexity regions. It means that you can get rid of it without fitness cost. So that's why they are not that important. So we can remove them or mask them. And after that, for each 3-word possibilities, for example, if K is 3, so K can be more than 3. But so basically, what are the possibilities? Word 1 is P, Q, G. Word 2 is Q, G, E, and so on and so forth. So after you have this list, you just search for the words in the database, right? So generally, so 13 is used by default. And after, for example, this is after you have a match and you extend it towards both sides, okay? So how do you do that? You just basically look at the BLAST algorithm, okay? So this is, for example, P and P is basically 7. If you just look at the BLAST algorithm, it's 7. This is also 7. You just extend it towards both sides. And this is L and V 1. Then you have F and V minus 1. Okay. Then you stop your scan, and this is your high scoring segment pick. Okay. And this is basically your sort of local alignment, but not necessarily. And we will come to that later on if necessary. So, BLAST algorithm, there is one important parameter that we should all know about, which is E-value. So what is E-value, right? So it is called expect value, and it's basically defined as the number of hits that you expect to get by chance only, okay? So if E-value is 1, for example, you expect to get at least 1 hit by random chance, okay? So there is no homology, so you expect to get 1 hit. So that's why the lower the E-value, the more significant your hit is. And it depends on the score. So what does it depend? So first, it depends on the score, right? And it is obvious. And it is negatively correlated with the score, obviously. If you have higher score, you have lower E-value. And there are 2 more parameters that we have to think about. One is the length of the alignment. And the second one is the length of or the size of the database. Okay, so imagine that if the database is large, then you expect to get random chance hits more, right? So that's why E-value really depends on the database that you are searching against. Okay, so we have one question. So why do we use other matrices? What are their advantages? Okay, so let's go back to that real quick. Okay, these ones, right? This is a very good question. So physical properties. Physical properties are not necessarily providing the entire information regarding the evolutionary history of sort of amino acids. So what do I mean? So sometimes, how do you capture physical properties first of all, right? So you have one value that summarizes everything. For example, the size of the amino acid is one physical feature. The charge of the amino acid is another physical feature. The polarity of amino acid is another physical feature. So if you have one value, which one is more important? So you can estimate that, okay, size is more important or charge is more important. But in a way, you have to summarize these physical properties in one value. And you may not be able to do that. Yeah, combination of them, right? So of course, combination, but you have to have a weight, which is more important. Or you can just take some sort of average. But evolution doesn't work that way. Sometimes, for some amino acids, charge is more important. For some amino acids, polarity is more important. For some amino acids, the size is more important. For some amino acid pairs, right? So that's why we don't know. How do we know? How can we understand it? It is not possible to test it in the lab as well. But what is great about this evolution is we have data available. Throughout millions of years of evolution, we know how likely for one amino acid to change or to substitute the another, right? So that's why we use mutation data rather than physical properties. Because physical properties are limited information. We can just guess but mutation data is statistics that we have. So that's the reason why it provides sort of much better scoring. So Shewal, does it answer your question? Great. Okay, let's move on. Okay, then for local alignment, you basically concatenate this high pair scorings into longer segments, and at the end we have your BLAST. So I don't want to go into detail regarding BLAST, but there are several options that you can BLAST nucleotide against nucleotide database, protein against protein database, nucleotide against protein database, or protein against nucleotide database, and finally nucleotide against nucleotide database, but just by converting them to protein sequence, which I will not go into detail for now, but if you have questions, I can answer them. Now I will stop this presentation, and we have to talk about multiple sequence alignment quickly. So bear with me. Okay. I'm sharing my screen again. Okay. Now, so far, we have talked about pairwise sequence alignments. Now, I think it is a good idea to talk about multiple sequence alignment because they are the basis of phylogenetic trees. So the assumption here is for multiple sequence alignment, first you have to have 3 or more biological sequences. And the assumption is you assume that they are homologous. Okay, so this is different than what we had with pairwise sequence alignment, because especially for local alignment, you don't necessarily have that assumption. You are just searching for homology, basically. Okay, and you don't have to have that assumption. For global alignment, you have to have this assumption. For multiple sequence alignment, it is like global alignment, right? But with more than 2 sequences. So that's why the assumption is that the sequences that you are dealing with are homologous. So they share common origin. So why do we need them? So firstly, if you are interested in discovering conserved motifs in a protein family, then you have to look at the sort of the entire protein family rather than only 2 protein sequences. So this is important because, as we discussed before, the conserved regions will give you some information regarding the important functional units of the protein. So multiple sequence alignment is a way to find that. And again, we will talk about phylogenetic trees. If you are interested in discovering or inferring evolutionary relationships between sequences, then multiple sequence alignments should be your choice. And also, it resolves some ambiguous situations that I will talk about. So it also helps correcting pairwise sequence alignments. How does it do so? So this is a nice example. Just look at the above. This is pairwise sequence alignment. Imagine that you are interested in aligning this above sequence with bottom one. Okay, so then this is the alignment. However, if you use more sequences, and if you do multiple sequence alignment, what do you get? Okay, so just look at here. This portion is not aligned with this part, okay, because they are not favored. However, if you use other sequences, you will see that this side is aligned with this side. How do we get that? Because you just look at the conservation of these green regions. So if they are conserved here, then they must be aligned with each other. So basically, we use the conservation information, just like in P and R here. So maybe R is not, but this is also seen to be sort of relatively conserved. So we use the information that we take from the other sequences, and it actually helps us to build better pairwise sequence alignments. Because here in pairwise sequence alignment, we are restricted or we are limited to 2 sequences only. However, for multiple sequence alignments, we can use the information that we can benefit from other sequences. And this improves pairwise sequence alignment as well. Okay? This is very important, and I think it is very useful for pairwise sequence alignments, but this is not the only use. So as I said, there are more uses of multiple sequence alignments. So how do we score them is the question. So there is one way of scoring is basically it is called sum of pairs. So what we do is we just, if you have multiple sequence alignment, you just compare for one sequence, for one position, what you can do is basically you compare sequence 1 versus sequence 2, 1 versus sequence 3, and sequence 2 versus 3, right? So after you have that, for every position, you can have a score. In this case, this is gap versus gap, gap versus C, and gap versus C again. So basically, you never score gap versus gap. It is 0 because it doesn't make sense to have any score between 2 gaps, because these 2 gaps basically are introduced because of this C, the insertion, let's say. Okay, so that's why comparing gap with another gap has no biological meaning. So that's why we have 0 for them. However, C versus gap has some sort of biological meaning, which is the gap penalty function. So now we can have 0 for gap versus gap. And this is gap versus C, it is 1G, and gap versus C, it is another G. So overall for this position, it is 2G, the score, right? So now you can do the same thing for the third position. T versus T is match, represented in capital M. T versus A is mismatch, and T versus A is another mismatch. Now we have 2 mismatches and 1 match. It's going to be the score for the third position. And you continue doing that, and at the end of the calculation, you have one score, which is called sum of pairs, basically. So normally I would have an activity here, but I'll skip that. Basically, all you need to do is, if you are given this +1 for match, -1 for mismatch, -1 for gap, so all you need to do is compare pairwise sequences for first position and second position and come up with the overall sum of pairs score, and you can easily do that. So it is the same logic with the dynamic programming that we have from previous lecture. So if you remember, we had 2D matrix, right? However, in this case, it is more than 2D because we have more than 2 sequences. So imagine that we had 3 sequences. So instead of rectangle, now we have a cube. And the same idea holds here. So instead of 3 neighbor cells here, now we have more neighbor cells with 3 sequences. And if imagine that, it is hard to imagine, but if you had the 4th sequence, then we cannot really imagine a 4-dimensional space, right? So how do you do that? Basically, we can calculate it, but the complexity is going to increase a lot. So how does it increase? It is actually n to the power k, right? Actually, this is also important. Each cell has 2 to the power k neighboring cell, and k represents the number of sequences, and n represents the number of cells. It means the length of the sequence, okay? So this is not a big deal, k squared. It's not a big deal in complexity. What is big deal is basically length as well as this to the power k. So it is also, I mean, this is not doable as well. So because if you have 1,000 sequences, then it becomes impossible again. So what I'm saying is, it is very complex when you want to do dynamic sequencing, dynamic programming. So again, this is only for 3 sequences. And how many possibilities we have? 1, 2, 3, 4, 5, 6, 7. So how many we had? 3 for 2 sequences, right? For 2 sequences, we had 3 possibilities. For 3 sequences, we had 7. So what is the pattern? It is 2 to the power n minus 1. So the complexity is to the power, sorry, k to the power k, right? So if we had four sequences, how many possibilities that we would have to consider the neighboring cells? Now it will be to the power 4 minus 1, which is going to be 15. So that's why complexity increases a lot. And I would like to show you how to decrease this complexity and how algorithms deal with that. So there are some sort of inexact approaches, okay? I will skip that and I will come to that star alignment. So star alignment strategy actually chooses some sort of a center sequence in the middle by just imagine a network, and in that network, basically the distance network imagine, and where do you start aligning sequences? It is a good idea to start with the sort of the center one, so that your multiple sequence alignment will be better guided. So what you do is, if this is the center one, so you basically start pairwise sequence alignment, and after you have your pairwise sequence alignment, then you merge them basically. So this is what you do. Again, details, I cannot really give you the details, but at the end of the day, you have your resulting alignment here. So because one problem of star alignment is once you open a gap here in pairwise sequence alignment, they have to stay as gap. So you cannot really do much about it. And this is your sort of resulting alignment. But the optimum alignment can be different than your resulting alignment. So that's why there has to be some sort of a refinement step here. So after you have your resulting alignment, there are ways to optimize it to get closer to the optimum alignment. And one option is called progressive alignment. Basically, in progressive alignment, what you do is you have a distance matrix. You generate the distance matrix first, and then you do pairwise sequence alignment, and then you merge your pairwise sequence alignment. So imagine the complexity now. So complexity was n to the power k, whatever. So the k was to the power k, so that's why it was highly complex. However, in progressive alignment, because we are reducing the search space by just clustering the sequences, now the complexity is reduced to the k squared or n squared, right? K squared times n squared is going to be your complexity. So what I'm saying that is your search space is now narrow and much narrower than the overall dynamic programming approach. But one thing that you have to consider is in progressive alignment is the profiles. So what is profile, right? So let's discuss what profile could be. Okay, I'm doing okay in time. So when I ask you, imagine a pen, okay? So what do you imagine? So what comes to your mind? So how do you imagine a pen? The first thing I think at least that comes to my mind is some sort of a stick, and it has a sort of sharp end, and probably it writes, but I don't really imagine that. But this is the picture that I have. And how do I generate this picture in my mind? So far in my life, I have seen many pens, right? I see a variety of them. So, but I have some sort of a general profile of a pen in my mind. And that profile actually represents the entire universe of pens. If this is the case, so I have one profile in my mind, right? Can we do the same thing for protein sequences? So can we represent the protein family or DNA family with a profile? Actually, we can. How do we do that? And there are ways of doing that, which we will not go into detail, but basically imagine a protein family with multiple sequence alignment. How can I represent it as a profile? So we can get statistics, right? For first position, I have 60% glycine, 20% alanine, and 20% arginine. I'm just making up. So this is one way of representing a protein family. And same thing can go to the DNA sequences. There are other methods like HMM, hidden Markov models as profiles. We can also have that. So why this is necessary is because now we are still doing pairwise sequence alignment like algorithm. However, this time we are aligning profiles because now you are interested in aligning many sequences, more than 2 sequences. And pairwise sequence alignment doesn't work because now you have profiles. How does it work? I will tell you. So this is how it works. So first, this is called cluster W, one of the most frequently used multiple sequence alignment algorithm. I'm not a fan of it, but I will give you how it works. So first, it builds pairwise distance matrix. There are ways of doing that. So after you have it, you can have some sort of a guide tree. So basically, we will talk about how you build trees. And then after you do that, you first align these 2 sequences with each other. This is just regular pairwise sequence alignment. So there is nothing fancy here. And then you just do this alignment. So how do you add it to this alignment is the question. So now here, after you generate your pairwise sequence alignment, you generate a profile of it. And you align this profile with this sequence, okay? And then you have another profile for this sequence, and you align it with this fourth. And at the end of the day, you have a profile representing these 4 sequences. And what we do here is we have another profile here. Now we can align profiles with profiles, right? It is just like pairwise sequence alignment, but this time, our entities are not necessarily sequences, but they are profiles. At the beginning, they were sequences. But after you have an alignment, now you deal with, you treat this clade as a single sequence, but one exception is that it's not a sequence, it's a profile, okay? I hope it makes sense. Let's see if we have any questions. Okay, no questions so far. Okay, moving on. Again, there are some problems of cluster W. So one problem is optimal alignment may not be found, and guide trees is derived from pairwise distances, and they are not much reliable. So pairwise distances are necessary because you have to have a tree, a guide tree. And how you do that? First, you have to have this matrix, which is basically pairwise distance matrix. However, this is not based on any sort of complex algorithm. It's probably Kramer based method. So, and also highly divergent sequences, this cluster W doesn't work well. So there is one strategy more, which is called iterative alignment. So basically, what you do here is after you have an alignment, there is sort of an iteration. So you remove one sequence, you align the rest, and see whether score is improved. And if it improves, you just keep doing that. So this is what you do. You have an alignment, you remove one of them, you do realignment and align the discarded one back and see whether the alignment score is increased. If increased, keep doing that. If not, if it converges, then stop, okay? So this is called iterative alignment, and it is also used frequently. I'll give you one example. So this is tea coffee, and similar strategy. First, you have to have some sort of clustering, and you have to have a guide tree. And after that, each pairwise sequence alignment is going to be represented as profiles, and this is a profile, and this is another profile. And then you align these 2 profiles with each other. And at the end of the day, you have your multiple sequence alignment. But the idea is similar to what we have. So in BLAST, we had sequence here in this matrix, sequence in one sort of axis, the sequence in other axis. Instead, here we have profiles, right? And you can do the same, you can apply the same strategy with the profiles. Now, the complexity is just as we had in pairwise sequence comparison algorithms. So that's why it is doable. With this matrix, we can actually generate multiple sequence alignments. So there are some template-based alignment methods which are useful at certain conditions. So what do I mean by templates? There are several templates that can guide your multiple sequence alignment. So these templates can be structures, can be profiles, or other things. But what is important here is you want to align these blue sequences with each other. But if you have a template aligner, so which is a sort of a guiding alignment, so you can use that. So let's say that they are well-established homologs. And if the well-established homologs, and if you know that they are homologous to these blue ones, you can use them as a guide. And this guide will help you to align these blue sequences with each other. And at the end, you can remove the templates and you end up with this sequence. So if this was a pairwise sequence alignment, then these blue ones would be aligned better than the pairwise sequence alignment result. So remember how multiple sequence alignment improved the pairwise sequence alignment, right? So this is the same strategy. Improve multiple sequence alignment with some sort of a template. So biological knowledge, you have to have that. And that biological knowledge will help you to have better and more promising multiple sequence alignments. One example. So let's say that you want to align this this green sequence with this red sequence. Okay, this is what you want to do. If you do that, there are two L's in this red. Which one will align with this L? This second one or first one is the question. If we know nothing but these two sequences, you don't know what to align, right? What to align. This L or this L should be aligned with this green L. If you have profiles that represent this green sequence, for example, this profile is the all homologous sequences to green, then this L is conserved. And we have another profile that represents the red sequence, then this L is not as conserved as this L. If this is the case, then you can, by just looking at this conservation, you can say that, okay, second L is most likely the homologous of this L rather than this first L, because first L is not as conserved as second L. The same conservation is in the first profile, so that this L should be aligned with that L, okay? So this is how homology-based template-based multiple sequence alignments can improve your multiple sequence alignment. Okay, so there are RNA-based templates. So in this case, what you are seeing is basically the hairpin structure. So as you might know, RNAs can form double or secondary structures. So how do they do that? Basically, pairwise complementarity between nucleotides, G and C, A and T, they are together. So with this one, then it forms hairpin structure, right? Secondary structure. So if you know that secondary structure is important for this RNA, you can use that as a template. How? Because you know that this and this actually form secondary structure, but nothing is important, but it is important secondary structure. I mean, the secondary structure is functionally important, then you don't really care about this alignment. Normally, you wouldn't align these two reds with each other because they look weird, right? They don't look alignable. For A's, you have T, for C's, you have G. However, in the context of secondary structures prediction, basically this part and this part actually perfectly form secondary structure. And if you know that, you can align these red parts with each other. Although they don't look to be homologous to each other, actually they are complementary. But what is more important is this green part is perfectly complementary to the red part, and same thing is for the above sequence. So this is how you can guide and align the middle sequence, which is the more important part, I believe. Okay, so if you didn't have this information, then it would be very hard to align this middle sequence with this one. And I think the final two tools that I will talk about is muscle and MAFT. And I have a schematic rather than verbal slide. So muscle and MAFT are frequently used tools, and they perform better than cluster W in most cases. So basically, what you have here is you have to have a guide tree first, because this is how you just merge these neighboring sequences with each other. So you start with unaligned sequences, and after that, based on the k-merge, so sort of subverts, by counting them, you basically generate a distance matrix. And after this distance matrix, you build a UPGMA tree, which we will talk about in the next lecture. And then you do progressive alignment. And after that alignment, now you can have better distance matrix. So this distance matrix was sort of an approximation. But after the first alignment, you have better distance matrix, therefore better tree, and you have another multiple sequence alignment, which is going to be better than first multiple sequence alignment. And after that, you basically generate a tree. However, now there is some sort of iteration here. You sort of cut the tree into two groups, and then you compute the profiles, and you do profile versus profile alignment, and you have another multiple sequence alignment. So as you can see, so first multiple sequence alignment is improved with better distance matrix. With second one, now we basically divide the entire tree into two. We have profiles and align profile with profile, and we have most likely better multiple sequence alignment. If score is better, then go with that and do the same thing. Go back and again generate tree and divide into two and redo the calculation and so on. If score is better, do again the same thing. If not, then the resulting multiple sequence alignment is going to be your final multiple sequence alignment. And this is a very nice strategy, and it's iterative, and it works well so far. And MAFT is sort of more accurate than muscle, but it is slower. So if the speed is not an issue for you, you can also use MAFT. Again, it uses the similar strategies like progressive alignment, reconstructing the guide tree, refinement methods, and so on and so forth, which you can look at it and understand the details if you like. And there's also MAFT homologs. This is very similar to template-based strategy. Basically, you just find some sort of homologs. You are not interested in those homologs. You are just interested in your sequences of interest, but you can get additional help from the databases to better guide your multiple sequence alignment. So these homologs, which are represented in grays, they better help you to align these red sequences. And after they are aligned well, then you can remove the gray ones, and you end up with red sequences which are aligned better than it would have if you didn't have these gray sequences. Okay. So what I'm saying here is there are some useful tools that if you're interested in multiple sequence alignment, which are basically Mega. Mega is user-friendly. It has an interface, graphical user interface. Most of the time we use command line versions of the tools, but Mega is a nice sort of tool for biologists, most likely. And JAView is a tool to visualize and edit multiple sequence alignments. So it is sort of interesting to have a need to edit multiple sequence alignment. Why in the world you would edit multiple sequence alignment? Because it is the output of sophisticated algorithms, right? However, life is not that simple. Most of the time, what you get from multiple sequence alignment algorithms can almost always be improved by manual correction and manual edits. And unfortunately, it requires some sort of experience as well. But you can just pinpoint what is wrong by just looking at the multiple sequence alignment. And JAView provides an opportunity to manually edit multiple sequence alignments. So maybe in one lecture, we can talk about how to do them later on. So I think this is it about me, but I can take questions. we'll have one more lecture together, which is going to be on phylogenetic trees. So unless you have any questions for multiple sequence alignments, let's give a break and come back in 15 minutes, which